we are going to talk for a few days, really, about meiosis. You'll see I've called this lesson 3A, and then tomorrow we'll look at lesson 3B, and then on Monday we'll build models that will represent meiosis and mitosis. So we're going to talk about this for a few days, and we're going to compare it to mitosis. So there's going to be lots of review built into that. So what's meiosis? Well, meiosis is something that happens in germinal cells. When we talked about mitosis, we said it happens in somatic cells. And I said somatic cells with body cells. Soma means body. Germinal cells specifically are the ones that will give rise to gametes. These are the cells that sperm or egg come from if we're talking about sexually reproducing organisms. So a germinal cell is the one that's going to make a sperm or an egg. So it's different than your regular body cells, bone cells, muscle cells, etc. Now, when we talked about mitosis, it was part of a somatic cell cycle. And so I would like to specifically point out that for germinal cells, it's not a cycle. They don't go through the processes and then just end back up at the beginning. It's a straight line path where once you're at the end, you're at the end. There's no going back to the beginning of this particular pathway. Now, the two things that are on the screen are telling you the two main purposes of meiosis. If I had to pick which one was more important, it would be the one I put as number one. So if anyone ever asks you what is the primary purpose of meiosis, it is number one, reduction division. So we got two words in the title, reduction, that means less, and division, so we're dividing. So we are still going to talk about cell division, right? One cell making more cells. But this time, we're going to produce cells that have fewer chromosomes, generally half, than the parent cells. So in humans, for example, regular diploid cells have 46 chromosomes, but gametes, the sperm and the egg, only have half, only have 23. So that is the main, number one, most important reason for meiosis. Now, because of reduction division, we can have number two. So that's why I said it's the more important of the two. Recombination is possible because of reduction division. So the products of reduction division, gametes, sperm or egg cells, have different combinations of genes. And when gametes combine, children, or offspring if we're not talking about people, are genetically distinct. So what we're saying is, by making these cells that have half the chromosomes that the parents do, when two of those haploid cells join up, a sperm and an egg join up, now you get a different combination of genes because half of them came from mom and half of them came from dad. And maybe you'll remember, it's in grade 9 where you talk about biodiversity and why that's a useful thing. You would have talked about it hopefully in Bio30E. But diversity is generally a good thing for a population. If every organism was identical, one thing could wipe an entire population or colony out. But by having variation, different organisms have different traits and can adapt to different things. Now, that is why it's such a big deal. Does anyone want to ask anything about those two reasons? So,
sorry, can I see that first?
oh, you sure can. So we're going to go through the phases of meiosis. And when you look at the names, you're going to be like, hey, this is the same as mitosis, because it does. It has the same names. Now, it doesn't mean they're identical phases, but they're similar. And so what I'm going to try to do is focus on the stuff that's not the same, so that you get an idea of what makes prophase 1 of meiosis, and that's not a typo. There should be a capital letter I there, which represents the Roman numeral 1. What makes prophase 1 different than just prophase of mitosis? Now, just to let you know where we are in a cell's life here, interphase would have happened right before this. And interphase would have looked the same as it did in a somatic cell cycle. So what will happen for these germinal cells is they'll go through interphase, they might even go through mitosis and interphase a few times, but at some point they'll branch off and they'll stop going through the cycle and start going through this meiosis pathway. So interphase, exactly the way we discussed it before, would be what comes before this. Now, here is something that is distinct. Unlike in prophase of mitosis, where there was just chromosomes, they're just hanging out. This time we have pairs of chromosomes. I'm gonna see if I'm using my finger. Oh no, that's not any better. Well, no, that doesn't look good. Pairs of chromosomes. That's different. In prophase of mitosis, I said nothing about there being pairs. There was chromosomes wherever, they were condensed, you could see them, but that was it. And these pairs of homologous chromosomes line up side by side. It's called synapsis. That's the name of the process. And so what it looks like when a pair of homologous chromosomes line up side by side is it looks like you have 4 pieces. And that's what my second point describes. A pair of chromosomes where each chromosome is actually 2 chromatids, right? Because we had interphase. In interphase, we go through the S phase where we replicate the chromosome. We make an identical copy that's attached by the centromere. And so when I take a pair of chromosomes, and each of those chromosomes has a chromatid attached, it's called a tetrad, because, oh gosh, I like this smart board. There, number 4. Forget the other parts, but tetra means 4. Maybe you remember that from chemistry stuff where you're naming things. Tetra is the prefix for 4. And it's because it looks like there are 4 pieces. So in my little picture down here, there are 2 sort of red rectangles and the letter T pointing to them. That's to try to highlight that there are tetrads there. In the top tetrad, there is a black colored chromosome with its chromatid attached, so it looks like a little X. Right next to it is a red chromosome. It's the same length, but it's a different color. And the different color is to try to illustrate for you that it doesn't have the exact same variations of genes, right? So it's the same length, centromere's in the same spot in the middle, but they're not identical. So those are homologous chromosomes. That's why they're different colors here. In real life, they're not different colors. I'm just using colors to illustrate that they're not identical. Just for saying's sake, let's say on all of these pictures, the black chromosome came from dad and the red one came from mom. It doesn't mean anything in real life, but just for these pictures, let's just say that's how it was set up. The second tetrad, you can see, has 2 longer chromosomes. One red, one black. The red one has an identical copy attached, that's a chromatid. The black one also has an identical copy attached at the centromere, also a chromatid. Those identical copies get made during the S phase of interphase. Now, that was a bunch more words. Anyone want to ask anything about those words so far? Okay, so we got a pair of homologous chromosomes. They make a tetrad. In that tetrad, there are 4 chromatids. Now, in the middle of our tetrad, we will have what we call non-sister chromatids. Chromatids that are homologous but not identical. It would be like one red and one black. So in the middle of the tetrad, I have a black chromatid right next to a red one. They are non-sister chromatids. And what can and often does happen is that the tips of those chromatids will cross over each other, literally, a physical crossover. We're not metaphorically trying to say something. If you look at the bottom tetrad, you might see at the very right-hand side, the red chromatid is sort of crossed over the tip of the black one. And then on this other picture on the side, I know it's red and green instead of red and black, but it's just a better illustration. It's showing you how when the tips cross over, like in this picture in the middle, they can actually exchange parts of their genetic information. So crossing over is a way that additional variation can happen. Variation is already happening because we're going to make gametes that are going to recombine, but this would be like an extra source of variation. What it means is the chromosomes that say I passed on to my daughter might not look the same as the chromosomes that I actually have because this crossover could have happened. Now, these pictures are using two different colors. What diploma questions have a tendency to do, just because they're cheap about color printing, is to use black and white. So you'll very often see one black, one white chromosome, and if crossing over has happened, you'll see that the tip of the chromosome will have changed color. That'll be your visual evidence. Now, depending on how much crosses over, this could be a few hundred, it could be a few million genes, like it could be a lot of genetic information. So that's prophase 1. The stuff I've listed here is what's different. The other stuff about prophase that you already know is still true. Nuclear membrane still is decomposing. Centrioles still moving to the poles. You can see them here on either side. They're labeled with a C. You can see the dotted line of the nuclear membrane. It's the black dotted line. That's still true. That's still happening. The chromosomes are still condensed so that I can see them. So all the stuff you already knew about prophase is still true. This is what's different. Okay. Questions? Comments? Okay, let's take a little body break because I want to see if I can figure out. All right. So we had talked about prophase 1. And like you saw in the video, interphase came before this. So we started out with duplicated chromosomes. They had their sister chromatid, which is an identical copy attached, and we had our tetrads. Next, we have metaphase 1. Metaphase 1 is similar to metaphase and mitosis. Stuff lines up in the middle, but there's a distinct difference. The difference is that the spindle fiber, which still attaches to each centromere, isn't going to guide each chromosome individually to the equator. It guides a tetrad, that whole group of 4, together to the equator. That's what's different. In metaphase of mitosis, they wouldn't have been paired up like that. They would have just been single X's in a line. But in metaphase 1 of meiosis, they are still in homologous pairs. That is distinct. Now the chromosomes, like I said, because they're lined up in homologous pairs, it means that one chromosome is on one side of the cell and the other is on the other side. And one of the things that she kind of alluded to in the video, but that I'm going to elaborate on, is that the assortment is random. We call this independent assortment. So in prophase 1, crossing over was a thing that happened that caused variation. In metaphase 1, independent assortment happens, which is another thing that can contribute to recombination and variation. Now, if you look at my picture, you'll see there's A and B. That's 2 possible ways that the chromosomes could line up in this cell during metaphase 1. Now, in these pictures, we're looking at a cell that doesn't have very many chromosomes. 2n only equals 4 here. We don't tend to use human cells as pictures for meiosis and mitosis because we have too many chromosomes and nobody wants to draw 46 chromosomes. I don't, you don't, you can't, there's too many. So we're using a smaller number here to make it simple. If I look at version A, notice that the red short chromosome is on the left, but the red long chromosome is on the right. They're on opposite sides of the equator. But in version B of what could happen during metaphase, the red chromosomes are both on the left. What we're trying to show you is that the chromosomes within their pairs, within their tetrads, can line up any random way. The red ones, which you might recall I said, we'll just assume came from mom, and the black ones came from dad. There's no rule that says all the red ones have to be on one side or all the black ones have to be on the other side. It's very random. And so we use the word independent there to try to convey the idea that the way that one pair is oriented doesn't affect the others. So in each pair, so a pair would be the two chromosomes plus their sister chromatid that are the same length. The chromosome of paternal origin means from dad, that's what paternal means, is on one side, and the chromosome of maternal origin, maternal means from mom, is on the other. But the orientation of one pair is independent of the others. So it doesn't matter how one pair is lined up, the next pair will line up randomly as well. So one time a cell might go through meiosis and all the red ones will be on one side. Another time it might be half and half. Another time it might be two red ones on one side and then all the red ones on the other side. So it's very random how the chromosomes line up in terms of which part of the pair is on which side of the cell. Now, does anyone want to ask any questions about homologous pairs, independent assortment, metaphase 1, just in general? Okay, now I'll just repeat something that I've said before. I will generally not require you to draw things, not on tests anyway, but you always can. If you find it easier to show me with a picture than with words what's happening, you can. But I will definitely show you pictures. That's something that will definitely occur. So you should be able to recognize, if I showed you this picture, that this is metaphase 1. And you might say, oh gosh, how would I know that? How would I know for sure that this is metaphase? Well, the chromosomes are in the middle. That's characteristic of metaphase. The spindle fibers have guided them to the center and are attached to the centromeres. That's characteristic of metaphase. Now, how would I know it's metaphase 1 of meiosis and not just regular metaphase? Well, they're in pairs. That's distinctive. And look at the tips of the chromosomes. You can see that there's a bit of black on the red one, a bit of red on the black one. It's showing you that crossing over has already happened. And so that's a clue that this is metaphase 1 of meiosis and not mitosis, because crossing over doesn't happen in mitosis. Now, if anyone wants to ask anything at all, yes?
In just regular metaphase, aren't they all in a straight line?
Yeah, so in regular metaphase, they'd all be in a straight line. Now, remember that the line doesn't have to be a horizontal line, right? So in these pictures, it's a vertical middle that we're looking at, and I picked a different middle on purpose just to remind you that when a cell is a circle, anywhere can be the middle. They're still in a line, but the line is going up and down this time instead of side to side. Anybody else want to ask anything there? Then we have anaphase 1. So in anaphase 1, like we saw in the video, we're going to pull things apart. So just like in regular anaphase of mitosis, the spindle fibers shorten and they're going to pull things towards the poles. But here's what's distinct. We are not separating those two chromatids. That's what happened during mitosis. Instead, we're going to take one of each of the homologous chromosomes with its sister chromatid still attached and pull them to each side. The sister chromatids stay attached. The centromeres have not yet divided, and they don't until we get to Meiosis II. And so she mentioned in the video that we watched this idea, and this is a very distinct thing. The thing that I always use to determine whether I'm in Meiosis I or II is whether or not the sister chromatids are still attached, because they stay attached all the way through the first phase of meiosis. Now, how do I know this is meiosis and not mitosis? Don't forget, in addition to what I just said, you can still see the evidence of crossing over happening. And the diploma artists have, in recent years, relied very heavily on that. They will show the evidence of crossing over very distinctly almost all the time when we draw pictures of meiosis, so that it's very clear that that's what happened. So up until this point, Anaphase 1, there are 4 chromosomes in this cell. Everybody agrees? There are 2 of each chromosome. There's a red and a black one of each length. So if I was counting the number of chromosomes in this cell, I would say it's diploid, and I would say 2n equals 4. Does anyone want to ask questions about that? Okay, because this is where something's going to change. We said that the number one reason that we need meiosis is for reduction division. We're going to make cells that have less chromosomes than the original parent cell. So when we get, oh, just kidding, I forgot I had this slide. When we get to telophase, that's going to happen. Now, the thing I have here represents yet another process that can cause variation. Although this one generally is not a positive or good thing. The first 2, crossing over and independent assortment, happen all the time to everybody. It happened when you were made, I'm sure. But nondisjunction is not a common process. It does not happen all the time. And the outcome tends to be negative if nondisjunction has happened. Now, nondisjunction is when chromosomes, like in Anaphase 1, or chromatids when we get to Anaphase 2, do not separate correctly. You already answered a question about nondisjunction. We just didn't call it that. It was the question that asked, hey, if a cell ends up having 92 chromosomes and the other one has none, when did this happen? It was on the worksheet from a couple days ago. Well, we knew that it would have happened in Anaphase. They didn't separate properly. Now we're just adding the name, nondisjunction. That's what it's called when the chromosomes or chromatids don't separate in Anaphase. Now, if you look at my little picture, it's just showing you like a little cartoony pow on the pair of chromosomes that's not being separated properly. And it's not the details I want to focus on, but on the outcome. Normally, when I'm making gametes through meiosis, I'm going to end up with haploid cells. And we know that haploid cells have N chromosomes. N is for haploid. But when nondisjunction happens and we're making gametes, we could end up with gametes that, if you look, they have N plus 1 or perhaps N minus 1. They're haploid plus 1 extra chromosome, or they're haploid minus 1 chromosome. Now, you've seen this before when we looked at karyotypes. There were some people whose karyotypes had an extra chromosome, and nondisjunction is just the name for what caused that to happen. Now, I'm going to describe what it means to have an extra or to be missing a chromosome on the next slide. But first, does anyone want to ask any questions about what nondisjunction is? Okay, so I'll just say it one more time. I showed you a weird thing that happens in each of the phases. Crossing over, independent assortment, non-disjunction. They tend to get grouped together in questions, like there'll be 3 of the 4 choices in a multiple choice question, because they're the things that cause variation and recombination. So, the consequence, which is what I wanted us to focus on, not the details of the process, is that we could end up with gametes with too many or too few chromosomes. If non-disjunction happens, and I end up with a gamete that is missing a chromosome, I call it a monosomy. Mono means 1. You should have 2 of each chromosome in the long run. But a person who has a monosomy, what that means is that one of their parents' gametes was missing a chromosome. So instead of getting 2 copies, one from mom, one from dad, they only got 1. One of their parents didn't have that chromosome in their gamete because of non-disjunction. There's a syndrome that exists called Turner's syndrome. Turner's syndrome, oh yeah, I'm not going to be able to, I'll write it on the board. Turner's syndrome, we would represent with an X and a 0. And you might even say, oh, that was one of the choices in a multiple choice question that we did, and I didn't know what that meant. In a person who has Turner's syndrome, they will have inherited just 1 X chromosome, but not a second one. It would mean that either their mother's egg or their father's sperm didn't have a sex chromosome in it because of non-disjunction. Now, you do not have to memorize the names of all the syndromes. I've said that before. But if I told you that's what Turner's syndrome is, you should be able to tell me, oh, I know what caused it, non-disjunction. And you should be able to identify it on a karyotype if we tell you what it is. Now, the other possibility from non-disjunction is that we'll make a gamete that has too many chromosomes. And then when that gamete fuses with another, and we have a child or offspring, there will be an extra chromosome. Trisomy is the word for it. Tri means 3. It's when a person has 3 of a chromosome instead of 2, like they're supposed to. Down syndrome was an example on that very first worksheet that we did. Down syndrome is trisomy 21. Chromosome 21 has 3 copies instead of 2. It means that either mom's egg or dad's sperm had 2 copies of that chromosome instead of 1, like it was supposed to. So when they join, the child ended up with 3. Now, someone will ask, what happens if you're missing 2 or you have 2 extra chromosomes? We're not going to talk about it because generally that's fatal. The embryo won't even develop. It's too big of a difference, and it would be not viable. So it wouldn't even survive, like the person wouldn't even be born with it. But people can and have survived missing a chromosome or having an extra chromosome. Now, does anyone want to ask any questions about that? There are lots of syndromes that have names for this, and I just want to repeat, you don't have to have memorized them. There's no point in the curriculum that says you are required to know by heart what each of the syndromes is, but you have to be able to identify them if we give you a description. Okay, so kind of like what we did in our karyotype assignment, I told you what it was. Down syndrome is trisomy 21, and you had to figure out if the person had it or not by looking at the karyotype. That's the kind of thing you need to be able to do. Okay, so far so good? So then we get to telophase 1. I was leading up to this, I was building up the excitement, and then we had to take a detour to non-disjunction. But telophase 1 is where we're really going to see number of chromosomes be different. Now in telophase 1, just like in regular telophase, there's pinching. We should get chromosomes unraveling and nuclear membrane forming. However, the cell, once it's done with telophase 1, is just going to hop into meiosis II. And some cells like to take a shortcut and just sort of jump right into meiosis II. And the telophase step is not super distinct. And I'll just sort of explain why, and then you'll see when I talk about the next phase. In telophase, the big thing that happens is the nuclear membrane forms and the chromosomes unravel. The next thing that's coming up is prophase again. What happens in prophase? The nuclear membrane breaks down and the chromosomes condense. Seems sort of silly to make something you're just going to break right away and to unravel something you're just going to condense right away. So there are some cells that will sort of hop over this step. They will still have to go through the pinching and the division that's going on, but you might not see this telophase step distinctly because the nuclear membrane might not reform because it's just going to get broken down, and the chromosomes might not unravel because they're just going to get condensed again. So that's what I mean by that first point. Now the cytoplasm divides, and in meiosis, the telophase 1 step happens almost concurrently with cytokinesis. So unlike mitosis, where like this happens, then this happens, the cytokinesis is happening in conjunction with telophase 1. So the chromosomes unraveling and membrane forming might not be visible all the time. What is really important, though, is that you are getting 2 daughter cells, each of which is haploid. So if I look at the left side where I have one of my cells and the right side where I have the other, how many chromosomes are on each side? Well, there's 1 red and 1 black on each side. There are 2 chromosomes. Sure, they're still duplicated. They have their sister chromatid attached, but there's only 2 chromosomes on each side. So remember before I said in anaphase, there were still 4 chromosomes, 2n equals 4. As soon as we are done with telophase 1, like once telophase 1 has completed, we call the cells haploid, and we would say they have n chromosomes because we've split them in half. The other thing that's important to note is that the chromosomes are not replicated before the next phase. So there's no interphase here. And sometimes people mistakenly think that every time you're done with telophase, you go back to interphase. Not for meiosis, because remember, it's not a cycle. Yep. Now, does anyone want to ask anything about telophase 1? So now we have meiosis II. And you'll see that I've got a little series of pictures there representing meiosis II. And meiosis II, if I were to describe what's happening in each phase, I would say the exact same words as I said for mitosis, except that the cells are haploid at the beginning. In meiosis, the cell is diploid up until the end of meiosis 1. Then it's haploid for meiosis 2. In mitosis, the cell's diploid, and it stays diploid. So other than the fact that the cell is haploid, and that's what we're dealing with, it looks exactly like mitosis. Nothing is different. In this little set of pictures, you'll notice here that the first circle, which I'm going to attempt to draw here, is sort of conspicuously absent. That's where prophase would go. And you might remember I said the cell kind of likes to skip a step if it can. Prophase would look the same as it does in mitosis, except that some cells sort of skip over it, because you'll remember in prophase what happens? The nuclear membrane decomposes, chromosomes condense. Well, if they didn't bother making a nuclear membrane and unraveling chromosomes in telophase, you kind of don't need the prophase for that to reoccur. So it'll look sometimes like the cell jumps over the first step and heads right to metaphase. And sometimes pictures that represent meiosis will do that because often the unraveling and raveling of chromosomes, the forming and breaking down of the nuclear membrane, it doesn't happen. So if I take a look at the pictures that are here, this picture is metaphase. Now this is Metaphase II, not Metaphase I. It looks exactly like mitosis, right? Got chromosomes lined up individually along the equator of the cell. There is a spindle fiber attached to each centromere. There are two pictures because remember, I made two cells in meiosis I. So at the end of meiosis I, I have two daughter cells, and each one of them is going to go through these steps. So that's why there are two copies of the picture. The next pictures are anaphase, and you'll look and you'll be like, that looks exactly like mitosis. In anaphase of mitosis, the spindle fibers shorten and they pull the chromatids to opposite poles. So if I was going to describe this, I wouldn't use any words that are different than how I described anaphase of mitosis. If I take a look at telophase, same thing is true. I got my pinching, looks like I'm getting two cells formed. Depending on whether telophase has just started or not, the nuclear membrane might look like a dotted line around the chromosomes. Oh, I wish the smartboard would take my pen where I'm writing it. Or it might not be formed yet, but there's that classic pinching. So, if meiosis II looks exactly like mitosis, how do I really know what I'm looking at? Maybe I knew how many chromosomes the cell had to start, and that could be my clue. But what if I didn't? What if I didn't know how many chromosomes the cell had in the beginning? How would I know this is meiosis? Well, I'm going to point out the best clue ever, the tip of that chromosome. The tip of the red chromosome is black. That tells me crossing over happened, and crossing over does not happen in mitosis. So, even though that picture of metaphase looks exactly the way mitosis would look, I know this is meiosis because I can see that crossing over happened. And like I mentioned, that seems to be the artists who draw pictures for diploma exams most favorite thing ever, is to show the crossover as the big clue. That's how you know whether it was mitosis or meiosis. Now, if you have a question,
Is there